In the previous video, we studied intercellular junctions, and today, we are going to understand cell adhesion molecules, or CAMs, in detail. Now, before you think this sounds boring, let me ask you a question. Why don't the cells in your body just fall apart? You're made of trillions of cells. What keeps them organized into tissues, organs, and systems? The answer is cell adhesion molecules. Think of them as the molecular cement, Velcro, and zip ties of your body. By the end of this video, you'll understand exactly what they are, how they work, why they matter clinically, and you'll have this topic completely nailed for your exams. Let's go. Let's start with the big picture. When you look at this diagram from Gray's Anatomy, you can see a typical epithelial cell, and on its lateral borders, you'll notice a series of junctions stacked from top to bottom. At the very top, near the apical domain, we have the tight junction, also called the occluding junction. This is the seal of the cell. Just below it is the zonula adherens, a belt-like junction that wraps all the way around the cell. Then comes the macula adherens, also called the desmosome, like a spot weld between two cells. At the base, anchoring the cell to the basement membrane, are the hemidesmosomes. And scattered on the lateral walls? Gap junctions, tiny channels for direct communication between cells. All of these junctions are built using cell adhesion molecules. Now let's understand exactly what CAMs are. So, what exactly is a cell adhesion molecule? By definition, they are transmembrane or membrane-anchored glycoproteins that bridge the intercellular space from the plasma membrane to form adhesive contacts. Let me break that down. Transmembrane, they sit in the cell membrane, spanning from inside the cell to the outside. Glycoproteins, they have sugar chains attached, which help with recognition and binding. Bridge the intercellular space, they literally reach across from one cell to the next, or from the cell down to the extracellular matrix. Now, the most important classification you need to know. CAMs are divided into two big groups based on whether they need calcium to function. Calcium-dependent CAMs, these include cadherins and selectins. Calcium-independent CAMs, these include Ig CAMs, which includes Nectins and Integrins. And here's a memory tip. Integrins are the only CAMs made of two subunits, an alpha and a beta subunit. Every other CAM is a single protein. Integrins are unique in this way. Remember that for your exams. Let's now go through each group in detail. First up, cadherins. The name comes from calcium-dependent adhesion, so the Ca is right there in the name. Cadherins are single-pass transmembrane glycoproteins. They have 5 heavily glycosylated extracellular domains, and these domains bind calcium. Without calcium, cadherins lose their shape and cannot function. So calcium is essential here. On the inside of the cell, cadherins have a cytoplasmic tail that binds to proteins called cadherins. Now, what are cadherins? Think of cadherins as the adapter proteins. They connect the cadherin to the actin cytoskeleton inside the cell. This connection happens at the zonula adherens. So the chain is cadherin, catenin, F-actin. That's how the junction is anchored. Now, how do cadherins actually stick cells together? They use what we call homophilic transinteractions. Homophilic means same binds to same. A cadherin on one cell grabs an identical cadherin on the neighboring cell. It's like a handshake where both hands are identical. After this transbinding, cadherins also cluster sideways on the same cell surface. This is called a cis interaction, and together they form a zipper-like structure that creates very strong, stable adhesion between cells. Now, different tissues have different types of cadherins. E-cadherin, found in epithelial tissues. The E stands for epithelial. N-cadherin, found in nervous tissue. N for neural. P-cadherin, found in the placenta. P for placenta. 2 more members of the cadherin family, and these are very important for desmosomes, are desmoglines and desmocollins. Now, where are cadherins found? In the zonula adherens and in desmosomes. And critically, where are they not found? In tight junctions and hemidesmosomes. This is a common exam question, remember it. Clinical significance. Alterations in cadherin expression in the epidermis cause conditions like blisters and ulcerations. A classic example is Pemphigus vulgaris, where autoantibodies attack desmoglein 3, causing desmosomes to fail, and the skin literally separates and blisters. This work on cadherins and the cytoskeleton was elegantly reviewed by Breher and Yap in 2013, published in Current Opinion in Cell Biology. They showed how cadherins cooperate with both actin filaments and microtubules at junctions. Next, selectins. Like cadherins, selectins are also calcium dependent, but here's where they become very different. Cadherins use homophilic binding, same binds to same. But selectins don't do that. Instead, selectins bind to carbohydrates, sugar molecules. This makes them a type of lectin, a protein that recognizes and binds sugars. Each selectin has an extracellular domain called the CRD, carbohydrate recognition domain, and the shape and binding ability of this domain depends on calcium. So what do selectins actually do? They are responsible for leukocyte homing, the process by which white blood cells circulating in the blood are guided to tissues during infection or inflammation. Here's the analogy. Imagine your white blood cell is a bus traveling through a blood vessel highway. When there's an infection somewhere, the endothelium, the lining of the blood vessel, puts up a slow down sign using selectins. The white blood cell slows down, starts rolling along the vessel wall, then eventually crawls out of the blood vessel into the tissue. This process is called extravasation. There are 3 types of selectins. L-selectin, found on lymphocytes, guides lymphocytes to lymph nodes through specialized vessels called high endothelial venules, or HVs. E-selectin, found on endothelial cells, expressed during inflammation to recruit neutrophils. P-selectin, found on platelets and endothelium, released rapidly from storage granules called Weibel-Palade bodies during injury, the first responder. Memory trick: L for lymphocytes, E for endothelium, P for platelets. The significance of leukocyte homing via these molecules was beautifully described by Gerard, Mushin and Forster in 2012, published in Nature Reviews Immunology, a landmark paper on how immune cells traffic through lymph nodes using HEVs and lymphatics. Now let's move to the calcium-independent group. First, IgCAMs, or immunoglobulin-like cell adhesion molecules. The name tells you the structure. They have an extracellular domain with immunoglobulin-like loops. These are the same kind of loops you see in antibodies. The number of loops varies between different IgCAMs. Most IgCAMs span the membrane normally, but some are attached to the membrane through a GPI anchor, a lipid anchor, instead of a transmembrane domain. Like cadherins, most IgCAMs also use homophilic binding, same grabs same, but without needing calcium. Inside the cell, the cytoplasmic tail of IgCAMs interacts with F-actin, and chirins, and spectrin, connecting to the cytoskeleton. They can also directly or indirectly bind growth factor receptors and control their internalization, so they have signaling roles beyond just adhesion. Different types are found in different tissues. NCAM, neural cell adhesion molecule, found widely on neural cells. Important in brain development. ICAM, intercellular adhesion molecule, found on vascular endothelial cells. VCAM, vascular intercellular adhesion molecule, uses heterophilic binding, meaning it binds a different molecule, specifically integrins. Now, nectins and nectin-like molecules, or NECLs, these are a subgroup of the IgChem superfamily. They have 3 extracellular Ig-like loops, a transmembrane segment, and a cytoplasmic tail. There are 4 nectins and 5 NECL molecules. They're found in tight junctions and zonula adherens. Here's the really important concept: the nectin-affidin complex. Nectins actually initiate the formation of the zonula adherens. Think of nectins as the scouts. They arrive first to establish cell-cell contact. Once that initial contact is made, cadherins are then recruited to the site. Then affidin and alpha-catenin interact with each other, and with F-actin through adapter proteins to complete the junction. So, nectins lay the foundation. Cadherins build the wall. This was comprehensively reviewed by Takai, Miyoshi, Ikeda and Ajita in 2008 in Nature Reviews Molecular Cell Biology, the defining paper on nectins, their homophilic and heterophilic interactions, and their role in contact inhibition. And now, the most unique of all the cell adhesion molecules, integrins. Why unique? Because integrins are the only CAMs made of two subunits, an alpha subunit and a beta subunit, forming what we call a transmembrane heterodimer. Different combinations of alpha and beta subunits give different integrins. There are 18 alpha subunits and 8 beta subunits known, and their combinations give you at least 24 different integrins, each with different binding specificities. Now, what do integrins bind? They recognize a specific sequence on extracellular matrix proteins, arginine, glycine, aspartic acid. In single-letter amino acid code, that's RGD. This RGD motif is found on fibronectin, laminin, and other extracellular matrix proteins. The integrin's extracellular head binds to this RGD sequence to anchor the cell to the matrix. But here's what makes integrins truly special. They work bidirectionally. Outside in signaling, the ECM sends a signal inward. The cell senses its environment. Is the matrix stiff or soft? What proteins surround me? And activates internal signaling pathways accordingly. Inside-out signaling. Internal cell signals change the shape of the integrin, opening it up so it binds its ligand more strongly. Classic example. When a platelet is activated, inside-out signaling flips the integrin alpha-2b beta-3 to its high affinity state, allowing it to grab fibrinogen and form a clot. So integrins are like two-way radio systems. They can receive information from outside and transmit instructions from inside. Integrins are found at hemidesmosomes, where they anchor epithelial cells to the basal lamina. Clinical example. In multiple sclerosis, T cells use the alpha-4 beta-1 integrin, VLA4, to cross the blood-brain barrier and attack myelin. The drug natalizumab works by blocking this integrin, preventing those T cells from getting in. Barczyk, Carrasito and Gullberg published the comprehensive integrin review in 2010 in Cell and Tissue Research, covering all alpha by beta subunit pairings, their ligands, and links to disease. Now let's look at what these junctions actually look like under the electron microscope. Here in the top left, you can see the tight junction. Notice how the membranes of two cells are touching each other. There is no space between them. That's the seal. No molecules can pass through that junction paracellularly. Top right, the apical junctional complex. This electron micrograph beautifully shows all three apical junctions stacked vertically, tight junction at the top, zonula adherens in the middle, and macula adherens at the bottom. You can literally trace the junctions down the cell border. Bottom left, a hemidesmosome. See how the intermediate filaments splay out from a dense plaque on the bottom surface of the cell, anchoring it to the basement membrane below. It looks like roots of a tree gripping the ground. Bottom right, a gap junction. This freeze fracture image shows the characteristic particle rosettes of connexin proteins, those tiny channels that allow ions and small molecules to pass directly between adjacent cells. Each of these images is a real-world confirmation of everything we've discussed so far. Let me now give you the cheat sheet, everything in one table. Tight junctions, use claudin and occludin, no cytoskeletal link. Calcium independent, most apical, key scaffold proteins ZO1, ZO2, ZO3. Zonula adherens, uses E-cadherin, linked to F-actin, calcium dependent, just below the tight junction, key proteins alpha and beta catenin, and offidin. Desmosomes, use desmoglines and desmocollins, linked to intermediate filaments, calcium dependent, basolateral, key proteins plakoglobin, plakophilin, desmoplakin. Hemidesmosomes, use integrin alpha-6 beta-4, linked to intermediate filaments. Calcium independent basal surface, key proteins plectin and BPG1. Gap junctions, use connexins, no cytoskeletal link, calcium independent, basolateral. Connexin 43 is the most common type. Screenshot this table. It will save you an exam. Let's now give credit where it's due, and understand why these papers matter. Breher and Yap, 2013, published in Current Opinion in Cell Biology. These authors showed that cadherins don't just physically link to actin, they actually regulate actin dynamics at junctions, and they highlighted the emerging role of the ARP2x3 complex as a junctional actin nucleator. They also brought attention to microtubules at cadherin junctions, something that had been underappreciated. Gerard, Mushin and Forster, 2012, published in Nature Reviews Immunology. This is the key paper on leukocyte homing. They explained how lymphocytes constantly recirculate from blood, lymph nodes, lymphatics, blood again, searching for antigens, and how selectins and HEVs control this traffic. Takai, Miyoshi, Ikeda and Ajita, 2008, published in Nature Reviews Molecular Cell Biology. The landmark paper on nectins. They characterized all 4 nectins and 5 necromolecules, showed their roles in contact inhibition of cell movement, and linked them to both adherins junction formation and cancer biology. Barczyk, Karasido and Gullberg, 2010, published in Cell and Tissue Research. The definitive integrin review, covering all 24 known integrin dimers, their ligands, the RGD motif, outside-in and inside-out signaling, and how integrin dysfunction leads to diseases from cancer to bleeding disorders. Let's connect everything to the clinic, because this is where anatomy becomes medicine. Pemphigus vulgaris, autoantibodies destroy desmoglein 3 in desmosomes. Result? Skin cells lose their grip on each other, massive painful blisters across the skin and mucous membranes. Leukocyte adhesion deficiency, LAD, a mutation in the beta-2 integrin subunit means white blood cells cannot adhere to the endothelium. They can't roll, they can't crawl out, they can't reach the infection site. These patients suffer from recurrent, life-threatening bacterial infections despite having plenty of white blood cells in their blood. Cancer metastasis. Loss of E-cadherin is one of the earliest steps in cancer spread. When E-cadherin disappears, cells undergo epithelial-mesenchymal transition. They lose their polarity, break free from the tumor mass, and become migratory. This is how cancer invades. Atherosclerosis. Inflamed endothelium upregulates E-selectin, ICAM1, and VCAM1, attracting monocytes to the arterial wall. Those monocytes become foam cells, and that's how plaques form. Multiple sclerosis. T cells use VLA4 integrin to breach the blood-brain barrier. The drug natalizumab blocks this integrin. It is one of the most effective MS therapies available. Epidermolysis bullosa. Defective hemidesmosome proteins like plectin or BP180 mean the epidermis cannot anchor to the basement membrane. Even minor friction causes the skin to blister and tear away, heartbreaking condition, and it's entirely a problem of cell adhesion. Alright, let's bring it all home. Point number 1. CAMs are transmembrane glycoproteins. They are the molecular glue that holds your tissues together. Point number 2. Cadherins are calcium-dependent, use homophilic binding, and link to actin via cadherins. Present in desmosomes and zonula adherens. Absent from tight junctions and hemidesmosomes. Point number 3. Selectins are calcium-dependent, bind carbohydrates, and guide white blood cells to sites of infection. L for lymphocytes, E for endothelium, P for platelets. Point number 4. IgCAMs and nectins are calcium-independent. Nectins are the scouts. They initiate zonula adherens formation before cadherins arrive. Point number 5. Integrins are the only heterodimeric CAMs, alpha plus beta subunits. They recognize the RGD motif. They work bidirectionally, outside in and inside out. Point number 6. When CAMs go wrong, disease follows. Pemphigus, LAD, cancer, MS, atherosclerosis, all have CAMs at their core. If this video helped you understand cell adhesion molecules, please give it a thumbs up, it really helps the channel grow. Subscribe to Gray's Gyaan so you don't miss any anatomy videos.